All right, we will go ahead and get started, and more folks can join for the main event when Mark starts talking in about 2 minutes. But first, just a brief introduction. My name is Nate Crosser, Startup Growth Specialist at the Good Food Institute, and I'd like to welcome you to the September 2020 installment of the GF Ideas Commercial Seminar Series titled The Business of All Proteins. As a reminder, this is part of a monthly seminar on business topics related to starting and scaling a plant-based, cultivated, or fermentation-enabled meat, egg, or dairy business. And you can sign up for subsequent presentations, join our complimentary series on the science of all proteins, or join the GF Ideas online community of over 2,000 entrepreneurs, scientists, investors, and subject matter experts at gfi.org/community. We have an exciting presentation in store today from Mark Warner on commercializing industrial biotechnology, particularly as it relates to alternative proteins. Mark has a tremendous amount of experience in this space as a former executive at both Impossible Foods and Solazyme, and as a consultant who has worked with over 50 companies as they scale their novel biotechnologies from benchtop to scale. We're very lucky to have Mark presenting today because he literally wrote the book on industrial biotechnology commercialization, which if you're interested in this presentation, you can order that on Amazon. That's a great resource. And this presentation really is relevant to all, I think, categories of alternative protein, but one in particular that it's really helpful for is fermentation. So that would be the use of fungi, bacteria, microalgae, and other microbes to either modulate plant proteins, create protein biomass, or create functional ingredients for plant-based products, such as Impossible Foods' heme, that gives it that bloody taste and appearance. And then if this is of interest to you, I do recommend checking out GFI's new report on fermentation as a key pillar of the alternative protein industry, which just came out yesterday. And you can download at gfi.org/industry to get an idea of the competitive landscape, investment trends, learn more about the technology, things like that in the report. So very brief housekeeping here. We have about 30 minutes of presentation from Mark, followed by Q&A for the remaining 20 to 25 minutes, which I will moderate. And please use the Q&A feature in Zoom. It's at the bottom of the ribbon, and I will save those questions for the end and moderate and ask the most relevant questions. Yes, a recording and the deck will be available both via email to all registrants sent later today, as well as in the agenda document where you can follow along our notes as we take them through the presentation, which I just dropped the agenda document link in the chat as well. And I think, yeah, the recording will also be put on YouTube. I think that's it, and I'm glad to pass it over to Mark.
Great, and you can see my desktop, Nate?
Yep, it's perfect.
Great, thanks. Appreciate the opportunity today. I'm excited to talk to everybody. This is a topic I'm very passionate about. I've spent a lot of time in recent years commercializing novel proteins and different food products, and would like to share my lessons learned on it. As Nate said, my presentation will go for about 30 minutes. There'll be Q&A at the end. Please, if you have any questions, drop a note in the chat. So with that, moving forward, I always like to start out briefly, just give you kind of my perspective. I think it's important when you look at information being presented to get a perspective of the person presenting it. As Nate said, I've spent time both at Impossible Foods as their chief engineering officer in the early days and senior vice president of engineering at Solazyme, commercializing some of the larger biotechnology projects in the space. But I also think it's important. I've also been a senior executive inside these companies. I was excited to hear there's a lot of founders on this call. What you're going to be faced with, and a perspective I feel I can bring, is representing both the technical side and the commercial side. Because as you look at the decisions you have to make on scale up and funding, a lot of it comes with discussions of valuation and things that are going to drive what you're doing. So I think that's important. As Nate mentioned, I've worked for 50 different early stage startups since I started my consulting group 5 years ago, and I've worked on over $1 billion worth of fundraising in the space. So with that, I'd like to move into some of the technical portion. I want to talk first about kind of our mission. My guess is that given the backgrounds of most people on this call, we share a similar mission in that I think the main goal most of us have is to get a wide scale replacement of animal use in commerce, replacing proteins and other products. And that's what a lot of this advanced biotechnology is focused at. It's hard to find a benchmark. I often use the recent A.T. Kearney study as kind of a stalking horse. They talk about replacing 60% of animal use for food, for meat by 2040. I think that's certainly an admirable goal. I'm going to talk about reasons why I think it's exciting, but that also on our current path, that's going to be a challenge. And there's some things I'd like to see maybe change or focus to get us closer. So let's talk, when I look at the space, about the good news first. There's a large number of really novel technologies out there. I meet with a lot of very early stage companies bringing these new technologies forward, and I'm always quite excited to hear what's coming, how broad it is, and how many problems it solves. The one thing I'd point out is, while there's a great buzz around a lot of these companies, I often see that there's not enough of them at really the later stage to where they're really producing a large amount of protein or near commercial scale. There's a lot of companies in very early stages. There's very few reaching that larger commercial scale. And I would really like to see that change. And I want to use a brief example of my time in biofuels. Like many that have been in advanced biotech for years, I started in really doing this in biofuels, and I learned a lot of things, both good and bad, from that experience. I often reminisce how the protein space is very similar at times to biofuels, in that there was a lot of buzz, a lot of funding, a lot of excitement about the space. But when you really looked at different times, there was all this talk of cellulosic ethanol and how it was going to replace gasoline. But when you really looked at the number of gallons produced, it wasn't that impactful on the total market. And it really took a lot of push and government driving force to get that change forward. And I feel like at times the novel protein space is similar. It's a lot of good buzz out there, but there's just not as many pounds of replacements out there today. And I want to focus on what we can do to change that and bring more of these technologies to commercial scale. So first thing I want to focus on, and this kind of overlays pretty well to if any of you haven't had the chance to see the fermentation report that GFI put out yesterday, this kind of overlays on that, in that although I define things a little differently, we have to look at the protein world in different sectors, because they're different technologies and I often see confusion between them. So I just wanted to briefly touch on them. You know, when I look at the range of products in the alternate protein space, I first start with what I'll call functional proteins. I think it's what the report refers to as precision fermentation. These are really the highly functional proteins, the impossible heme, the dairy proteins that the Perfect Days and the Clara Foods are making. These are things that aren't just a protein. They don't just have nutritional value. They have a function. Best example is, think of an egg. Why do you cook with an egg? Because it's a liquid at room temperature. When we get it to a certain temperature, it gels, it forms a structure. That's part of the reason you put it in cakes and other things. Then when I look at things like lower functional proteins, I think of things like Corbion's whole algal protein or Nature's Find microbial protein. These are what I would consider lower functional proteins. It doesn't mean they're bad. They're made in larger quantities. They don't have necessarily as much functionality to them, but they do often have the nutritional value. They often bring structure with them. So it's a different type of these fermented proteins. Then we can get away from fermentation and look at things like the agricultural derivatives. Think of like Hampton Creek's Just Scramble product or Beyond Meat Burger. These don't necessarily come from fermentation or synthetic biology. They're isolates of current agricultural proteins, but they do bring some functionality and also the nutritional replacement. Then there's the emerging technology of cellular meat. That's a little more of the advanced phase of meat replacements, companies like Memphis Meats, little more on the cutting edge. The main points I want to make are twofold here. First, we need all of these. If we're going to replace proteins at large scale, we need all of these. The second thing is, The points I'm going to talk about what makes a technology scale up successful is really true for all of these. So I would encourage everybody to, when you're thinking about this, don't focus on just your technology, focus on really kind of the general bigger principles of what makes scale up successful. So I want to move into talking about specifically scale up and what I consider scale up. And one thing I like to talk about is, let's talk about the goals and things of each stage. I think people often move through these stages without focusing enough on what is their real goal. So let's start at the pilot stage. This is generally proof of concept. You're at this point just trying to show you can make the protein you say you can make, and that it's in some order of magnitude, maybe the cost you think you could make it at, but it's really just making enough of it to show you can make it. Proof of concept. I do want to note most of my examples in here today are going to be regarding fermentation. I will tell you, when I give costs and different things, if you're talking agricultural derivatives, it's probably on the cheaper side of the range. If you're talking about things like the emerging proteins like cellular meat, it's probably on the higher end of the range. But again, the principles are the same. For fermentation, we're probably talking in the range of 2 to 1,000 liters. You're really only going to be making at most probably a kilogram a batch. A pilot plant, order of magnitude in industrial biotechnology, probably in the range of $1 million to say $5 million. And this is going to be all of an R&D expense. Again, the main thing you're trying to show here is really proof of concept. So let's move into what I would call the demonstration scale. This is a larger scale. You're going to make a lot more products. So instead of just proof of concept, you're going to use this usually for a couple goals. You're going to use the larger amount of product, one, to give to customers to be able to do things like sample testing. You have to seed the market enough. Think of something like you're making a protein for a granola bar or a protein bar. They're going to want to get, hundreds of kilos of it, most of the CPG companies, to make a bunch of sample products, send it out to focus groups, and really determine if they have a need to buy this and potentially fund the project. You also need to get enough engineering data to be able to design a bigger plant. So there's multiple goals here. Typically in the fermentation world, the scale of fermentation is in the range of probably say 5,000 to 100,000 liter fermenters. You're going to be making metric tons of material per batch at this scale. Order of magnitude costs for a demonstration facility, probably in the range of 50 million, maybe down to 25 million. I think one point I like to make here, and this is the relation between the technical side and the commercial side, is these are almost always unprofitable and bring very little revenue. I see a lot of early stage companies that are often optimistic and believe they can generate a lot of revenue at demonstration scale. Unfortunately, I don't see it happen that often. And this is important because if you're planning on that revenue as part of your scale up and funding plan and it doesn't come through, and I can tell you as a former senior exec at these companies with a fair amount of equity interest, you can paint yourself into a corner where you need to raise money at a valuation you're not happy with. So I would encourage people not to be too optimistic about revenue at the demonstration scale. Make sure you have enough funding in your raises to get through this scale without revenue. Hopefully I'm wrong. Hopefully you can generate more revenue and that gives you upside, but don't let yourself get painted into the downside corner. So let's look at what I would call first commercial scale. This is the first of a kind facility for a new commercial technology. This is the first time you're really hoping to be profitable. It's really what I would consider commercial proof of concept. Generally, these plants, the first of a kind, has to be equity funded. Your hope is to prove it out so that later facilities can be debt funded. That's usually the longer term goal. And this is really to show your technology works and make enough product to be out in the market in a stable way. In the fermentation world, usually these are full commercial scale fermenters, which are in the range of 200 to 500,000 liters. You're making tens of metric tons, potentially even up to hundreds of metric tons per batch. Commercial scale facility is often in the $150 to $400 million range. And I said, this is really the first time you're expecting to be profitable. I want to focus on the line on the bottom. This whole process is meant to de-risk the technology and your whole commercial business. The further you get along this process, the more real data you have, and the more you're able to de-risk the technology. So now that I've given kind of that framework of what commercialization and scale up looks like, let's start at the beginning. Say we want to make a new novel protein for food and look at specifically what does that look like. So I like to say, start at the end and work backwards. This graphic here is pretty much your typical fermentation-based process to make a food protein. I've probably dealt with 20 different companies that have a basic configuration like this. This is kind of the basics of most biotech-based proteins. It uses aseptic aerobic fermentation. After fermentation, assuming it's a secreted protein, the cells are removed with a distack centrifuge. That broth containing the target protein is then purified initially with a microfiltration, which is going to remove all particles and components above a certain kilodalton size range. Then there's going to be a backstop ultrafiltration step where the media and salts and things can be removed and the protein is held, and then it's spray dried for a final product. Very common commercial process. The question is, how do you take your brilliant idea you have that's on the bench and scale that to commercial scale? So the first thing I want to do is drop in the typical equipment I would see if I go into most biotechnology companies at their bench or small scale pilot. You can see these dropping in. First is probably a 2 to 5 liter benchtop fermentation system. Second is a benchtop batch centrifugation system. Third is a small pilot microfiltration system. Fourth is a benchtop ultrafiltration system. And the last one is a small benchtop spray drying system. These are all very representative of equipment and technologies I've seen used at various scales. The thing that's important here to understand is these will give you good basic proof of concept, and I'm going to give some more detailed examples of this, but they aren't always fully representative of the equipment you're going to get at larger scale, and you need to understand that going in to be able to de-risk it. The other thing I want to focus on is the difference in scaling up a unit operation, which is the individual fermentation, centrifugation, spray drying, versus the overall process. Many people in the industry are very focused technically on one, maybe two unit operations, but as you really start marching towards commercial, you need to focus on the overall process. And very commonly, for example, fermentation, you may need to run fermentation in a manner that gives you a productivity that may not be your best productivity. Maybe it's only 95%, but it gives you some other trait that lets the downstream recovery be more efficient so that the overall combined process, in this case of these 5 unit operations working together, is the best overall process you can put into the market. So in talking at this, I've come up with a way, and if you end up getting my book or looking at some of the publications I've published specific articles on this, I want to talk about how I look at this scale up process and consider the risk between phases. So I took those 5 unit operations and I represent them here in a block flow diagram of the 5 different unit operations. And then I'm going to talk about at each scale, pilot, demonstration scale, and first commercial. I'm going to talk about those unit operations, and I'm going to talk about 2 factors that relate the pilot to demonstration and demonstration to commercial. The first is the arrow on the far left. It's what I would call the scale-up factor. How much bigger is the demonstration facility than your pilot facility that's generating the data? In this case, I'm saying the demonstration facility is 185,000 kilograms per year. The pilot is 2,340, so that's roughly a 79-to-1 scale up between pilot to demonstration. The second factor is how much data, how much hours of operation do I have at, say, the pilot scale that I'm going to use to make my decisions on the design of the demonstration scale. Generally, when we do that, we like to get 1,000 hours at each scale for scaling things up. It can't always be done, but that's the target. On scale up factor, the first item I talked about, you'll often hear in the industry, people like to have something like a 10-to-1 scale up maximum. I can tell you that's generally what bank funding requires. Equity funded projects, I typically see maybe 50, 75 to 1 because the equity groups are willing to take a little more risk, but you need to understand that scale up factor. The other thing I want to talk about briefly is the types of unit operations. Discussing so far, I've basically assumed the unit operations are the same at pilot, demonstration, and commercial, and that's not always the case. And I'd like to use a centrifuge as an example. If you looked at the picture I put in that first drop-in, it was a benchtop centrifuge, which is a batch system. Those can often be very efficient. If you run them long enough, I can get 99.5% removal of many of the microbes by spinning it down on the benchtop. When I move to a larger scale and use something like a continuous distack centrifuge, I can often get a lower efficiency, say 95%. So what that means is two things. First, I'm going to lose 5% that I'd been getting previously, but it also means my downstream unit operations could change a little. The microfiltration system at the benchtop is now seeing 5% more organism coming into it, so it may change a little. These are important to understand where you change a unit operation or where it could impact things downstream. It also brings up a lesson I've learned, frankly, the hard way is the further you go down, or excuse me, the further you go upstream and make a major process change, it can impact everything else downstream of that. And you have to really be cautious of that when you're doing scale up. So let's move into what I would call some practical insights. A few things here. Make sure you understand your financing upfront. And the reason I say this is, you heard me talk about the 10-to-1 scale up that's required by debt financing. The equity financing will often go maybe 70-to-1. What's important is, I see this happen a lot where companies will assume they can get debt financing, but yet they're planning on doing a 50 or 60-to-1 scale up. They get very far along in the process only to find out they can't get the debt financing they planned on. I would tell you either of those routes are acceptable. Just make sure your plan for financing matches your plan from scale up and that you understand that at the beginning. Another thing I want to talk about is, as you look at, if you're forced into having to do some of these larger scale up factors, where I talked about doing something like, 75-to-1, you'll notice that's on a rough plant basis. That's overall plant. There's ways you can mitigate that. In the case of fermentation, what often happens is the plant may be a 50-to-1 or 75-to-1 scale up, but when you do your fermenters, maybe you don't go to the largest fermenter you could. You go to a fermenter that's only 20 times bigger, but you have multiple units. That is a way of mitigating some of this risk. You often hear people refer to it as scaling out instead of scaling up. That can be done, and I've done it. The only thing I would warn you is there are operating cost benefits to fewer pieces of larger equipment. So if you're going to take that approach, just realize that having more pieces of equipment is likely going to mean you're going to need more operators, so it could be more operating expense. One thing I didn't really talk about earlier and I should have focused on more is when you're doing these 5-unit operations and generating your data, it's critical that you have full integration, meaning from your your inbound feedstock to your final product, and that represents what you're planning on doing. This has all the recycle streams and things included. And the reason I say this is very often, for reasons of cost, companies will ferment at one site, then maybe hold it in a tank for a week and ship it to another site for some processing, and maybe ship it to a third site for spray drying. I'm not going to say I haven't done it, I have, but it can change the streams, and you don't always get the same recycle information to where when you build the full-scale commercial plant, it can be more of a problem than you think it is. So something to keep an eye out for. Finally, hours of operation. You heard me talk about the goal to have 1,000 hours of operation for your data set, and also to go no bigger than a 10-to-1 scale up. I can't tell you, there's not a lot of good in third-party industry guidance here. I can tell you my own personal experience. I'm much more willing at my own risk to go with a larger scale up factor, say 50, 75 to 1 doesn't worry me, as long as I've got good representative, fully integrated data, and say maybe 1,000 hours of it. Much riskier to me is trying to minimize the scale up factor, but using data that is not fully integrated or representative. So let's talk about economic implications for a minute. And this is another reason I like to focus on this, is the theme I talked about earlier. When you have your whole scale up plan, understand the data you need to get at each stage, but what are the costs at each stage? So you can look at this chart here, and it shows basically the unit manufacturing cost for proteins at pilot, demonstration, and commercial scale. And you'll notice a couple things. The price per unit drops dramatically as you get much larger in scale. And I can tell you, having scaled a lot of processes from pilot to commercial, I can tell you that's a true effect. I mean, believe it or not, it doesn't take that much more labor to run a 500,000 liter fermenter than a 5,000 liter fermenter. Certainly not 100 times more labor. So you'll see, because of that, the major individual cost components, the two shades of green, labor and overhead, are the two things that as a portion of the unit cost drop dramatically as you get to larger scale. The thing I want to point out is really, again, focusing at this demonstration scale. Pilot scale has a very high cost per kilogram. It's a number that can often scare people when they hear, hundreds of dollars per kilogram, but you don't make a lot of it, so it's not as big a deal. Demonstration scale is where it's still a relatively high cost, but you're making a lot of the material, and this is when you can generate a lot of negative costs. So you want to be really focused on your demonstration scale to really understand the information you need to get and generate at that scale, so you minimize the time there, because it's very often one of the larger components of your scale up cost. Having been inside of many companies, the point where I can reach positive cash flow is always the near-term goal. You want to make bottom line profit also, but the point where you can stop pulling in equity to continue your research is really a key milestone. I'd like to briefly wrap up with a couple notes here. And these, again, are kind of some of my key lessons learned. First, never forget, when you're a food venture, even though you're using biotechnology, you are a food company first and foremost. I see a lot of people in the biotech space that like to consider themselves a biotechnology company first and want to change the way food is made and sold. What I will tell you is that can be a big challenge. If it's something you really need to do, and I'll use the example of, and Good Food Institute has fought some of this battle, the naming of things like the word meat and cellular meats, that's something that's seen that has to change, that there's a reason to really push back hard. My concern comes more in the way food is manufactured and tested is often different than biotechnology, and the person who comes out to inspect you, being a food inspector, won't always understand biotechnology that well. You can cause yourselves a lot of problems, and it's often, quite frankly, an easier path to success to make sure you just understand how food is made and sold and try and fit into that if you can at all first. Full integration being a key, I talked about. Look for footprints along the journey. A lot of these technologies are somewhat similar. It's often things like the key organism and maybe the protein that are a little bit different, but a lot of the key unit operations are the same. I had a mentor at one point that used to always tell me, shamelessly steal others' good ideas. I'm of course not talking about intellectual property or trade secret material, but there's a lot of good lessons learned and best practices out there that can help you along the path. You don't have to rediscover everything yourself. So keep a lookout for good ideas. Don't be against using other good ideas outside of your core technologies to move things along. Go big or go home. This really focuses on that cost slide where I talked about the unit cost difference at very large scale. There are economies of scale, and they're real, to get to very large scale. That's why there usually needs to be a push to move down that commercialization path as expeditiously as you can, just because the real point that you're most likely going to get cash flow positive and start making money is really not to that very large commercial scale. Data quality over quantity, you know, you want to generate a lot of data, but make sure it's really quality. And I talked already about the funding source. Want to wrap up here. First, thank everybody for participating. We had a pretty good turnout today. My contact information is here. Please, if your question does not get answered during the Q&A, don't hesitate to drop me an email and reach out. As Nate kindly noted, I have published a book that summarizes a lot of this. You will find it on Amazon, or if you go to the resources page of my website. The book is really a compilation of about 30 different white papers or publications I've made over the last 5 years. You'll find most of the source documents there if you'd prefer to find them there as opposed to getting them through Amazon. And with that, I will pass it back to Nate for the Q&A.
Thank you, Mark. That was fantastic. And we've got about 25 minutes for Q&A here. So excited to do a deep dive into some questions. And everyone feel free to put your questions in the Q&A box and I will ask them. We already have a few keyed up. The first is actually a couple of questions kind of combined about timing. Could you maybe talk about what is the typical time from conception to commercial scale, and then what amount of time is typically spent at each of those three phases?
And the answer's twofold. You have to look at the construction number and how much data you need to generate. So I actually tried to put this in the slide, but it became a little too busy. If you ask me for this protein process I've talked about, what's the back of the envelope? Pilot scale, usually 6 to 12 months to build that. Demonstration scale, usually in the range of 12 to 18 months. And commercial scale, probably in the range of 24 to 30 months for a commercial scale fermentation facility. So if you add all those together, and I want to do it in my head, you're going to get like 3.5, 4 years. And I'll be honest, I see a lot of business plans that show that, but they often don't give time to generate data. Think of that 6 to 9 months for pilot plant. Say I'm fortunate and I can get that plant built in 9 months. I may need to run that for 6, 12 months to get the data I need before I can build the demonstration scale plant. I might be able to do some early engineering along the path, but that's the other thing you have to really look at is how much time do you need to generate after you get something up and running to get enough data to be able to design that next phase.
Great, thank you. Another couple of questions that we could maybe combine. Actually, no, we'll just talk about the CMOs. How do they fit into this? Contract manufacturers, do you recommend using one, and at what stages and under what circumstances? And maybe explain how that works.
Sure.
If people aren't familiar.
So when I came into consulting 5 years ago after leaving Impossible Foods, I had spent most of my career designing and building facilities, and that's what I assumed most people were going to hire me for. And the first year, I think I did 2 contract manufacturing searches. Last year, I did a dozen of them. And the reason is because you always fight the last battle. Biofuels overbuilt capacity, so there's a hesitance to build fermentation capacity. So there's been more of a lean to go to contract fermentation. And I will tell you up front, if you can find the right site and it works for you, it's very often a quicker and less costly route potentially. The problem is there's so many startups coming in, the world of CMOs is getting a lot smaller. So many of us have kind of forecasted kind of a crunch coming of limited CMO capacity. So what I advise most of my clients is you really have to keep both options open, because generally, even if you think you're going to go with a CMO, you're going to be asked how much would a plant cost and how long will it take. So I would tell you to keep both options open. Most of the companies today are leaning towards using CMOs. I expect that's going to shift back over the next year or two with more companies looking to build their own capacity.
Great. Then a little bit earlier in the production process, there was a question about working with universities on the pilot scales and whether there's enough capacity for that, and maybe just if you have general advice for working with universities.
Yeah, I've also done work on some companies looking to build some what I would call pilot scale facilities similar to universities. So I've done some benchmarking of them. There's, I'm in Texas now, but I was in the West Coast. There's a national lab, the ABPDU in Berkeley, that a lot of companies use. There's Iowa and Iowa State, I think University of Georgia. There, most of those have in the range of a 300 to 1,000 liter fermenter. I've had clients that have used all of them. There's certainly good options. MBI in Michigan has been an option too. They're certainly out there. They also have been harder to get into. It's just like everything else. It's going to be quicker than building your own. But I guess what I would tell you is make sure when you do that initial assessment, if you think you're only going to run for a month or two at one of these CMOs or even the pilot CMO, it's always going to look cheaper to use the CMO and not build your own. The problem is many of us have found out the hard way, 2 months can become 6 or 9 months, at which point it would have been cheaper for you to build your own. The other thing is, especially when you get to demonstration scale, while CMOs can be beneficial, I've managed multiple demonstration scale facilities, and it's important to understand anytime you use someone else's facility, you're somewhat limited in the work you can do there. They're going to try and meet your needs, but you can't always run the plant the way you want. One of the benefits from having your own pilot or demonstration is you can often run it your own way. But I tell people, this is the other thing I tell people, a lot of people want to build a demonstration scale plant. And I say, that's great, but building a demonstration scale plant is like getting a puppy. It's not a decision for today. It's something you're going to have to feed and care for many years. The problem is, if you want to use this demo plant for a year, you either then have to lay all the people off, which is going to cost you a lot of money to rehire them later, or continue to pay those salaries. So this is where you need to look at the short-term versus long-term benefits.
Yeah, just I guess while we're on CMOs, quick question. Do you sacrifice economies of scale by using a CMO in any way?
No, I mean, I've used CMOs up to 500,000 liters. Now, when we use the word CMO, and this is probably one of my next white papers, I've written about CMOs, but I need to talk a little more. CMO is a generic word, but the options, it's kind of like the fermentation report you guys put out yesterday at Good Food, kind of explain different portions of the industry. Let me take that word CMO and break it into categories. When I think of the big manufacturing companies, Cargill, ADM, that do large scale fermentation, to me, that's large scale, long-term manufacturing. When I heard CMO, that generally is a shorter term, a few batches, maybe 6 months or a year. It's often related, comes out of the pharma industry. It's usually a little smaller scale. Those are, and often it's people that are more willing to do scale up work. Because a lot of some of the big players, if you come to them and want to run for, 4 or 5 batches, a lot of the big manufacturing organizations don't want to do that. But as far as a practicality, I've been anywhere from 2 liter to 500,000 liter fermenters at CMOs and everywhere in between. So there are options out there. I do want to do a quick aside. to tell you if you hire me or another consultant to do a CMO search. I always like to tell people what the answer is probably going to be before you hire me for a search. The answer is probably going to be, if you're going to be over 100,000 liters and making a food product, the answer is probably going to be in Europe. It's not going to be in the US. There's a lot more facilities and capacity in Europe than there is in the US. And people are often surprised they don't have as many options closer to home, unfortunately. I'd sure love to see that change.
Yeah, thank you. So a question here on forecasting. Is it possible to reliably forecast what your costs per kilogram are going to be at commercial scale when you're, say early in the pilot scale?
Yes. I've been involved in doing techno-economic model from benchtop to taking that same process to full commercial scale. And I can tell you, I love to say it is possible as long as you're honest with yourself at the beginning about the assumptions. There's, I'll talk about the things that have surprised me over the years. Generally, you know your raw materials. Generally, the fermentation scales pretty well. Fermentation scales from benchtop to pretty large scale for many of the fermentations pretty well. The downstream is often a bigger problem. So a couple lessons I've learned. So many people in the industry come from advanced biotechnology. They're really fermentation people. There's often an underestimation of the complexity and cost of the downstream. In most of the plants I've been involved in scaling up, building and operating, it's almost $1 billion worth of projects. I can tell you the downstream usually costs more to install and is more complicated than the fermentation. And people don't often believe that going in. So I'll tell you, I see more problems generally in missing the targets on the downstream recovery. Now, again, I'm talking for things like some of the more complicated processes I've shown. If you're making like a corn type product where it's fermentation and spray drying, I'm not saying it's not complicated, but there's less steps. It may be easier, but the very complicated processes, it is more complicated, but it's been done successfully.
Yeah, and speaking of those slightly less complicated processes, could you talk a little bit more about how say a biomass fermentation would look different than creation of a functional protein, and maybe roughly what the different cost profiles are on a kilogram basis?
Sure. And they're dramatically different for two reasons. So first, we're using all these different informations, precision and biomass. Really what it means is when I grow the organism, am I getting just a small enzyme or protein out of it, or am I getting the whole cell? In the case, so if you think about that, you grow this cell and you get a protein coming out. That protein's probably maybe 1/10, 1/20th or more or less of the cell. So its first thing is in volume. This is why those precision proteins, the heme and other things are usually pretty expensive, because you have to grow this whole biomass, but you're only using a portion of it. Where in the case of the whole cell proteins, you're using the entire biomass. The whole cell, or what you call the biomass fermentations, are generally a little quicker than the precision fermentations. They may or may not be less complicated, but it's the downstream that's the dramatic difference. You may go from 4 or 5 steps of downstream to 1 or 2 steps. So, I mean, the price difference is huge. You can get, tens of dollars to low hundreds of dollars per kilogram for many of these precision proteins, where many of the whole cell proteins they're hoping to have for $1, $2, $3 a kilogram. Very dramatic cost difference. And that's why I like to tell people, I'm not saying one's better or worse. The precision proteins are then added as a functional ingredient to a much lower cost, say like, agricultural material to make a higher value product. Both are valuable.
Yeah, absolutely. Great point. So we have a couple of questions here around financing.
Mm-hmm.
Generally, the difference, the practical difference here between equity and debt.
Mhm.
Someone's asking, why would you do equity when you could do debt? So maybe talk about how that's
Oh,
not often an option. And another person opines that VCs are not investing in steel. So how do you, solve the problem of getting a VC to invest in your steel so you can get to the point where a banker will help you build your facility?
it's a great question, and it's probably one of the least understood. So it's the thing I'll say, and people think I'm joking when I say this. Difference between the debt and the equity people is the debt people need to be confident they're going to get paid back. They're lending you money at 8-9%, and they want to be absolutely confident your process works, that it's going to work, and their cash flow they project in the model is going to come back. By contrast, the equity people are hoping for transformational change, a 10-to-1 markup or a 10-to-1 escalation factor. A VC can get 2 or 3 big hits out of 10 companies and feel like that's a good hit rate, where equity can have a 10% total underperforming, and that's a problem. So I don't think people understand. I've been involved in a lot of big equity and debt financing. When you debt finance a plant, the first thing you have to understand is that plant is no longer yours. That plant's the bank's. And when you send a bill to a customer, the money goes to the bank, who takes their share out first and sends you any residual if it's left. So I don't think people always understand. I agree that for dilution reasons, you're always going to want to get equity. I would point to the biggest players in the space. Look at all these big fundraisers that a lot of these companies taking things to commercial scale. The reasons they're raising hundreds of millions of dollars right now is because the majority of them aren't funding their projects with equity, unfortunately. And that's one thing through this conversation I really want to see change, because we're not going to hit those big numbers of replacing animal proteins unless we bring equity in. There's not enough, bring debt in. There's not enough equity out there to fund the number of plants we need.
Yeah, yeah, absolutely. We need a lot of debt in
Yeah.
play here. So there's a couple of questions on regulatory, and because we're talking
Yep.
about food, maybe you could just briefly overlay the grass or the food safety relevant
Yep.
topics, in the commercialization process. When do you need to be thinking about that? If you've run an analysis at pilot scale, do you need to do it again at demonstration? Obviously you would at commercial. It
Yeah.
just maybe overlaid that a little bit.
So I on these plants I've built, I've helped coordinate them through grass, which usually means I know enough to be dangerous. So I'll start there, but I've been through the process many times. Generally, you have to generate, once you have your process developed, usually it's 3 to 5 batches where you generate at least 1 kilogram of material. You have to run a bunch of toxicology tests. There's generally 2 paths, generally regarded as safe. It's a government, I call it an approval, but it's a weird approval. You submit your information, and there's 2 pathways. What's called self-affirmed, where you get some experts to look at it and you yourself basically say, we've determined it to be safe. Or you submit that to FDA and they never approve it. They just quit asking questions. And the point, they have no further questions. They send you a letter saying that, and you'll see companies publish the, hey, we got a no further question letter, which is implied approval. So it's kind of 50-50. Some people are able to generate enough material at the pilot scale for fairly simple processes. The only thing I'll tell you is make sure, if you think your process is going to change or the unit operations are going to change, I've also seen people have to redo it. So I see it more commonly run at demonstration scale than on pilot, but either can be done. Generally, I tell clients, you'll hear 6 to 12 months to do it. I would tell you 12 to 18 months. The main question is the protein you're making similar to any protein people are eating now? And is the organism you're using being used to make food today? That's a way you can gauge the risk.
Yeah, that's really great. So we've got about 5 minutes more on the Q&A. So we'll go into a rapid fire grab bag of
Okay.
questions that didn't really group. So one is, what are the considerations when deciding where to put a facility, say in India versus the US? And what kind of cost difference maybe does that make?
Um, Generally, the biggest thing you look for any plant is feedstock. Assuming you're, and I'm talking most fermentations here, sugar and energy are the two largest feedstocks. So if it's going in the US, it's almost always going to go in the industrial Midwest. Um, India, I've dealt almost exclusively in the US and Europe and South America. I've been involved in projects in all three. There is a look at Asia. The main thing I would tell you on building a facility in Asia is most, and I'm not trying to justify it, it's just the case. Most US CPG companies won't buy food out of Asia. Now, someone's going to say, hey, I'm talking India, not China, because of some of China's issues, but it unfortunately gets painted with a broad brush. So if you're making it to serve the Asia market, great. If you're planning on building a facility there to bring to the US, you should make sure with your clients they're really willing to take it from there. But as of today, you don't see as many food products made in Asia like you do things like amino acids and other things.
Okay. Similarly, say you've got your pilot facility in New Jersey or whatever.
Yep.
Um, are you going to want your demonstration and your commercial facilities nearby, or does it really not matter?
You're going to want it there, but generally the economics of demonstration scale, it's one I've always say I always like it. It's seldom been the case. I would worry especially when you do the first commercial, I would worry about where the most profitable place is, not near your demonstration, necessarily near your demonstration facility. Very often at demonstration scale, you may find a fermenter at a CMO and put a downstream there or something like that. Where a commercial facility, you really need to make sure your economics work, and the sugar and the power are going to be the two biggest items.
Great, thank you. There is a question on alkali acid precipitation method, which is used at large scale in practice. Do you have any comments on that?
I don't, unfortunately. It's not something I'm familiar. If I could do, I'm looking at the things in the side notes too. Two quick things. Melly points out there was a good seminar and I watched it. I think it was the last one on debt financing. I would encourage people interested in that question to go back on YouTube and look at it. And I've seen a couple people pop up and ask if I'll share the presentation. We are going to put a YouTube out, but if you want the specific presentation, drop me an email and I'm happy to send it to you.
Great. I guess one more question. This says, due to the high speed of scale up, companies generally work on high production strains while generating data at, say, a pilot scale. What's your input on the amount of data generation needed when only replacing the strain, but the process remains the same?
You know, and this is another one I've learned the hard way. Unfortunately, the answer is it depends. People in the synthetic biology like to believe they're just going to make a couple tweaks to the organism and nothing else is going to change. I've had cases where we moved into demonstration scale and find out the organism was secreting a polysaccharide we didn't expect. One affectionately got referred to as the dead body strain because the smell was so odorous we couldn't handle it. Those are things you don't realize till larger scale. They are the minority. This is always, there's no clear yes or no answer. It's always a risk decision. I'm used to pushing forward aggressively and just having to deal with 10% of the time. And most of your investors in the space are going to tell you that. You know, if you're telling us it's a 10% problem, march forward and mitigate it when it happens.
Yeah. Well, Mark, that's great. And my brain has grown 3 sizes today. This has been incredibly helpful. I think we'll pop off a little bit early so we don't get kicked off by a webinar that we've got in a few minutes here. Mark, this has just, been really great. I know you shared your contact information. Would highly encourage anyone reaching out to Mark who wants to dive deeper into these topics, or to GFI if you want to talk about some of the funding sides of things, those kinds of questions. Also, we next month, kind of continuing in the same vein, we have a presentation from Jordan Stowe at CSC Leasing about the kind of tactical getting the equipment into these production facilities and how to get those financed. So we'll continue our our theme of scale up into next month. And we hope you have a really productive month until then.
Thank you.
Yep. Bye, everyone.